Hey everyone, today I want to talk about chapter 6, which is on the visual system, and we'll get started. So, first, want to be able to make sure that you know how to explain how the pupil and the lens can actually affect the image that falls on the retina. Now, before we can do that, we have to start at the very beginning of vision, which is light. No species can actually see in complete darkness. I know sometimes we think that that's the case, but the reality is that some animals are actually better at low light vision. And you can think about rattlesnakes, for example. They can actually detect infrared radiation, which lets them hunt at night. Now, light itself can actually be thought of in two ways. They can be thought of as tiny particles of energy. Those are called photons, or as waves of electromagnetic radiation. And every single thing that we see starts with these little packets of light entering the eye. So essentially, you're looking at an object, light is being reflected, and packets of light are entering the eye. So now that light has entered the eye, how does that actually get focused so that we can make sense of what we're seeing? That is where the pupil and the lens step in. Once light enters the eye, the pupil and the lens actually take over. Now, if we think of the pupil, we can think of it like a doorway, and it's actually opening wider in the dark and it's closing down in bright light. And the lens is like a built-in camera lens that's focusing the light on the retina. Now, there are also these tiny muscles that are called ciliary muscles, and they adjust the shape of our lens. And the process of that is called accommodation. And that's what allows us to quickly switch from reading a textbook up close and then looking across the classroom. Now, you've probably noticed that some animals' eyes look different. Before we get to that, I want to go through and just show you a little bit of an image here. Now, one thing that I want to talk about specifically is the iris. Now, sometimes, as I mentioned, if we're in the dark, right, we actually it gets wider, our pupils get wider, they dilate, and then sometimes they constrict, meaning they're getting smaller. Now, the cause of dilation is that the muscles of the iris, which is the colored part of our eye, relaxes to widen that pupil, right? And then it's how that's controlled is actually done through the sympathetic nervous system. If you remember from the sympathetic nervous system, that's kind of our aroused state. Maybe it's fight or flight, right? And it means that it allows more light in so you can see better in the dark. So this often happens when we're excited, when we're tired, when we're maybe nervous, aroused, stressed. It can also be triggered by some drugs, so including hallucinogens or stimulants. Now, the pupils, as I mentioned, also constrict. They're getting smaller. Now, the cause is due to the muscles of the iris contracting, and that's what caused the pupils to narrow. Now, this is controlled by our parasympathetic nervous system, which is kind of our rest and digest state. And when the pupils are constricting, it lets less light in to protect the retina in bright conditions. Now, you can think of this very similarly as when you step outside and it's super, super bright and you're coming into a more dimly lit room, it takes a second for your eyes to adjust. So that is essentially what's happening. And so it is associated with calm, with relaxation, even focusing on nearby objects. And it can also be triggered by certain opioids and certain medications. And if you could really just think of our pupils in any way, I would say the best analogy is like they are the emotional windows of the nervous system because they're adjusting to both our environment and our state. I want to go back to the question though on animals. So, why do some animals have eyes on the front of their heads and some have them more to the sides? Now, the answer in this actually tells us a lot about survival. So, predators like humans, lions, owls have forward-facing eyes, and that gives us depth perception, okay, which is super important for hunting. Prey animals, rabbits, deer, they have eyes that are more on the side. And that gives them a wide field of view so they can actually spot danger coming from almost anywhere. So, pretty fascinating. Now, once light passes through the lens, it actually lands on the retina. And this is the brain's little movie screen at the back of the eye. Let's see what's going on there. So we're going to talk a little bit about the structure of the retina. We're going to name some of the cell types that make up the retina, and then we'll talk about visual transduction. So once light passes through the lens, it lands on the retina. And that's the layer of cells at the very back of the eye. Now, the retina is actually packed with very specialized cells, and these are called receptors. There are rods, okay, which help us see in low light, but only in black and white. It's non-color. We also have the cones, which allow us to see color as well as fine detail. So these signals actually get passed along to bipolar cells, and then they go to the ganglion cells. Now, there are other cells, so horizontal and amacrine cells, and they play an important part too because they're helping fine tune the message. The retina you can think of as the eye's first brain. It's already doing some of the processing before signals actually even enter the eye. And that creates the first layer of visual processing. But the retina itself doesn't just capture light, it actually translates it into neural signals. Let's explore how we can do that. Now, there are some fascinating features here. There's something called blind spots, and the blind spot is actually the area where the optic nerve leaves the eye, and there are no receptors there. But what ends up happening is our brain fills in the missing information from the surrounding area. So you don't notice that there's a hole in your vision. Then we have the fovea. And this is a tiny spot in the center of the retina that is responsible for our sharp and very detailed vision. And that's why when you're reading, you move your eyes across the page. You're bringing each word into your fovea so you can see it clearly. And for those of you that were in class today, we talked a lot and actually did a blind spot exercise where we covered an eye and actually looked at, two kind of bar graphs that were next to each other. And we played with the distance in that to see at what point does that blind spot come into play. And for most people, it's about 5 inches in front of their face. So I want to, again, let's go back to rods and cones. Let's compare what each system is good at. Now, rods are super sensitive. They actually, like I said, help us see in low light, but also they're only in black and white. The cones are less sensitive, but allow for that fine detail and that rich color. And that's why stargazing relies on our rods, but reading this slide is dependent upon the cones. So they specialize in different types of visions. And rods, like I said, stargazing, they're great for night vision. And then cones need more light. So once we have this information and it leaves the retina, where does it go? So we're going to follow the pathway into the brain. And I do want to just really quickly show you a figure of the rods and cones. You can see the rods are in blue. They look a little bit more straight, and the cones look a little bit cone shape, and those are pictured in red here. Now, I want to talk about the eye movement because our eyes are in continual motion. And they make up a series of what are called fixations, and we have about 3 of them per second, and they're connected by the saccades. And those are rapid movements between those fixations. Now, the saccades are like bullets. So once it's on its path, there's really nothing you can do. Eye movements are essentially keeping the visual image in continual motion on the retina. And the importance of this movement is illustrated by the fact that stabilized retinal images disappear. And most visual system neurons respond to change and not steady input. So we talked a little bit about the action potentials and the excitatory postsynaptic potentials and the inhibitory postsynaptic potentials. And it's no different. Our neurons are firing. There are action potentials taking place constantly. Now, what we perceive at any instance of these eye movements is the sum of the input that's been received during the last few fixations. And this is known as temporal integration. And temporal integration of retinal images explains why our visual images are very detailed, why they're colored, maybe why they're wide angle, despite the very small size of our fovea. And it also explains why things don't disappear when we blink. So transduction is really just the conversion of one form of energy to another. And the first step in visual transduction is conversion of light to neural signals by those rods and cones that I talked about. Now, transduction by rods is very well understood, and there is a pink substance in the rods called rhodopsin, and light is absorbed by rhodopsin, and essentially it bleaches it, meaning it is removing its color. And this bleaching occurs because retinol and opsin, the two components of rhodopsin, separate in light. Now, the bleaching reaction that takes place actually hyperpolarizes the rods. Thus, the bleaching reaction is actually transducing light to a cascade of these like intracellular events that result in a neural signal. So when the rhodopsin is totally bleached by intense light, these rods lose their ability to absorb light and transduce. However, in the dark, the retinal and the opsin are reunited, and that rhodopsin regains its color and its ability to absorb light and transduce. Now, there is some strong evidence that the absorption of light by rhodopsin is the first step in what's called scotopic vision, which is the correspondence between the absorption spectrum and the scotopic spectral sensitivity curve. And so we'll continue to learn more about this. Now, I think it's also important to talk about the retina geniculate striate pathway. And the primary visual cortex is called the striate cortex. And in one of the first classes that we had, we talked about the Golgi stain and we talked about the Nissl stain. And how we discovered this striate cortex is because there was a stripe that was seen in a Nissl stained cross section. And that stripe is composed of the terminals or the buttons of axons from the lateral geniculate nuclei. Okay. Now, we said visual information is traveling from the retina, from those rods and cones. It goes to that geniculate, which is located in our thalamus. And then the striate cortex, which is in our occipital lobe. And that is our primary visual cortex. So visual information actually takes a very well-traveled route. I mentioned from the retina through the lateral geniculate nucleus of the thalamus, and then into the primary visual cortex, which is also called the striate cortex. So notice how the left and the right fields of vision actually are crossing over. Your brain is essentially weaving two slightly different images into one coherent picture. Pretty cool. Now, here's the fascinating part. Information from the left side of our vision goes to the right side of our brain and vice versa. So your brain is taking these two slightly different images and combining them into one seamless picture. And once the retina has done its job, the information actually travels much deeper into the brain. And we can talk a little bit about this, but we have the primary visual cortex that I mentioned located in the occipital lobe, and that is receiving most inputs from the visual relay of that nuclei that I mentioned, the geniculate nuclei of the thalamus. Once it is receiving the inputs there, it comes up to another level called the secondary visual cortex, and that is located in the prestriate cortex, and it's receiving input from the primary. So it goes primary, secondary, and then it goes even more, and it goes into the visual association cortex. And that visual association cortex is receiving input from the secondary visual cortex, and those areas are the inferotemporal cortex and the posterior parietal cortex. Those are the names of them. Now, the visual association cortex is also receiving not just information from the secondary visual cortex, but also from secondary areas of other sensory systems. And this is just an image of the visual areas of the cortex. So I know it's hard to see some of this text, but at the top you have the posterior parietal cortex, which is this orange. And let me get my little laser here, but it's going to be this right here. You have that pre-striate cortex that I mentioned. It's this blue right here. And then you have the primary visual striate cortex. So this is kind of your primary, this is your secondary, this is the visual association. And then you also have the inferotemporal cortex, which I mentioned right here. So what happens when there's damage to the visual cortex? Like, what does that look like? How does that impact our vision? When damage happens to the visual cortex, people actually can lose parts of their visual field. And sometimes they don't even realize it because the brain is trying to fill in some of these missing spots. So Carl Lashley, a famous neuroscientist, actually mapped out his own migraine-induced blind spots. And he described how parts of his vision would just disappear temporarily, which honestly is a reminder of how fragile the system can be. And so there are scotomas, and that is areas of blindness in the contralateral visual field due to damage in the primary visual cortex. And it is really plotted by a parametry test. And many patients with large scotomas, I'm sorry, as I mentioned it, are unaware of them. So, for example, patients that have, the complete destruction of the left striate cortex cannot detect stimuli in their right visual fields, right? Yet when they stare at a face or even half a face, that falls entirely in their left visual field, right? And then they report seeing an entire face. That phenomenon is called completion. Now, amazingly, when forced to guess, some brain damage patients can actually respond to stimuli in their scotomas. So they can, let's say, grab a moving object or they can guess the direction of its movement, all while claiming to see nothing, which is again fascinating. But this is called blind sight. And blind sight is thought to really be mediated by visual pathways that are not a part of this retina geniculate striate system. So, for example, one hypothesis out there is that the retina geniculate striate system mediates pattern and color perception, whereas a system involving the superior colliculus and the pulvinar nucleus of the thalamus mediate the detection and the localization of objects in space. Now, I will say, we talk about specialized damage. There are some very specific conditions that emerge from it. And one of the most studied ones is called prosopagnosia. And this is the inability to distinguish among faces. It's also known as face blindness or facial blindness. Now, people with this condition cannot again, recognize faces, even the faces of family and close friends. And this is caused by damage in the ventral stream of the brain, where visual information meets the areas that process object recognition. Now, here's something wild. So, even though they cannot consciously recognize faces, their bodies often respond unconsciously. So, for example, the skin conductance, and we talked about this in class last week, but their skin conductance actually changes when they see a familiar face, even if they say, I don't know who that is, right? Their brain on some level knows, even if they don't. So again, most widely studied form of visual agnosia. And then there's achinotopsia. Now, achinotopsia is the deficiency in the ability to see movement progress in normal, smooth fashion. So this could be an example of somebody with normal that can see smooth. If you're walking towards me, I can see those movements smoothly. If I were to have achinotopsia, it might feel a little pixelated or like it's glitching, kind of like my chair is here in the back, right? And it's a little bit more broken up. It's not a smooth, fluid movement. Now, it can be triggered by a couple things. So high doses of certain antidepressants can actually cause it, but it's often associated with damage to the medial temporal area, or the MT is what it's called, which is near the junction of the temporal, the parietal, and the occipital lobes. Now, sometimes the MT is also called V5 or MTV5. There are four really lines of research that have implicated the medial temporal area in the perception of movement. So one of them is that patients with damage echinotopsia tend to have unilateral or bilateral damage to movement. It's also measured by functional MRI, so fMRI, and the activity in the medial temporal area increases when humans view movement. Okay, so we know that too, that when humans view movement, there is activity in the medial temporal area. Now, blocking activity of the medial temporal with TMS can produce motion blindness. So that's another area of research. And then electrical stimulation of the medial temporal induces visual perception of motion. So all of those have been able to support the medial temporal area as being damaged. That is what it's affecting that, right? So we're able to look at this research and say, yep, we're supporting the fact that it's probably damage to the medial temporal lobe or the medial temporal area. And this image is just both of these conditions, and it shows just the lateral view of the brain. And you can see here on the left, we have what's called the fusiform face area, FFA, and the occipital face area. And that's associated with the prosopagnosia. And then damage to the area medial temporal is right here, and it's marked in red. And it's associated with echinotopsia. Now, the FFA and the OFA, you can't really see it. And the reason you can't see this in this figure is because they actually lie on the ventral surface of the temporal lobe and the occipital lobe, like respectively. Okay, so a little bit can't see it in this. And then the only other thing that I will say from a kind of vision deficiency is color blindness. And color blindness usually results from missing or malfunctioning cone types. Now, the most common is red-green color blindness, which makes it really hard to distinguish between those colors. There are videos out there, here are some links, but you can actually go on and look at tons of videos of this. And it's pretty fascinating to see. And different types of color vision deficiency, there are different types of tests that can actually show you that. So one of the most common for like the red-green color blindness is a test where you actually have, let's say, plates or little circles, and you have dots within them. And then you may have a number in there, like the number 74, the number 65 in one of those colors. And if somebody has a color vision deficiency, they will not be able to see the number. And so every type, whether it's a red-green deficiency, a blue-yellow deficiency, all of that have their own tests that can actually check for that. And again, there's even, I think, a lot of online color blindness tests that can help you. The only other thing I will say about color blindness is that there's definitely a genetic factor to it. So I know in one of our first classes, we talked about evolution and genetics. And so it is actually more common in males, and that's because of the X chromosome. So you're going to see probably more males diagnosed with color vision deficiency, more so than females. And that is all I have for you today. I hope this was helpful, and we will continue to dive into different sensory and perception processes next week.